our BPN Dual Virtual Seminar Series. Today we have the pleasure of having Dr. Yoland Smith, who is a professor at the and co-director of the Yerkes National Primates Research Center and Department of Neurology at Emory University in Atlanta. Dr. Smith received his PhD in neuroscience from Laval University in Quebec, Canada, in 1988. And after postdoctoral trainings at Oxford University and Johns Hopkins, he became assistant professor in the Department of Anatomy at Laval University. But in 1996, he joined the School of Medicine of Emory University, where he is currently professor and vice chair of the Faculty Development in Neurology and chief of the Division of Neuropharmacology and Neurological Disorders at the Yerkes National Primate Research Center. And Dr. Smith is a member of the Emory Udall Center of Excellence for Parkinson's disease and has published over 250 peer-reviewed publications on the pathophysiology of brain networks in Parkinson's disease and related movement disorders. He is the senior editor of the European Journal of Neuroscience, and he's deeply involved in graduate education as teacher, mentor, and principal investigator of the NIH T32 training grant that supports the Emory Graduate Neuroscience Program. So thank you very much, Yoland, for being here today, and we look forward to your talk. Thank you.
Okay, thank you very much. I think it's a great pleasure for me to be here. Thanks for the invitation. Always great to share some of our work, and particularly with students and future students who are interested in learning more about neuroscience and research. So this is really a great opportunity that, and I hope that you will at least get the feeling about what we are doing in our laboratory and give you an ideas about the kind of questions that are of interest in relation to Parkinson's disease. Please, if there is anything really I know that it's a very diverse group and people have different background. And so I just want to make sure that everybody understand and can follow the story. But if there is anything that is really seems to be off track and I somehow did not explain clearly enough and people get disconnected, please just let us know, and I'll be happy to stop and just really kind of re-explain it. And there might be things that are just not clear. So just please don't hesitate to ask question. Just want that you understand where we're going and what kind of the take-home message of this story. Okay, so let's get going with this. Then, as the title said, I'm going to talk about thalamic degeneration in Parkinson's disease and the potential impact of that on early cognitive impairment. So there's a lot of information here. So the thalamus, for instance, that some of you who don't necessarily do a lot of neuroscience may not know about. There is Parkinson's disease that we'll talk about, and we'll talk about also cognitive impairment, that is changes in cognition that are indeed affecting Parkinson's disease patients. So we'll see a little bit about what that means and how really what are the network of the brain that might be affected in mediating these problems. So as mentioned, I'm located, my laboratory is located at the Yerkes National Primate Research Center, which is on the Emory University campus. So the Yerkes Center is one of the 7 national primate research center in the US. There are 6 others, in this spread across the country. And so the idea is that really we are here, my lab is here because I'm using non-human primates in my research on Parkinson's disease. So this is what I've been doing for the past, 20, really almost 30 years now. So, and I'm using non-human primate, and you will see a little bit about some example of that during my talk. I'm also part of the Department of Neurology, as mentioned. I'm involved a lot in the neurology department. I'm not a neurologist. I don't necessarily see patients. I'm a neuroscientist, but I'm very much involved and in close relationship with my colleague in neurology. And as mentioned, also, I'm involved very much in mentoring junior faculty in neurology to help them advancing their own career. So what you see here, the picture that you see This is the front door of the Yerkes Primate Center on the Emory campus. And here is an overview of the Emory campus per se, which is, as you can see, very beautiful at this time of years in the fall. Lots of wood, lot of trees, and this is a characteristic of the campus, but also of Atlanta itself. It's a very green city, a lot of trees everywhere in the city. And here is my research team. These are the people who are doing the work in the lab. In fact, really, it's a makeup of a technician, lab manager, postdoc, graduate students, undergraduates. So it's a rich makeup, but these are people really who do a lot of the work that I'm going to talk about today. Okay, so let's get going. So we'll so what I'm going to talk about, or as you mentioned here, as I mentioned, we talk about the thalamus, and you see here a box diagram of some neural circuits that exist between the cortex, here the cerebral cortex, the basal ganglia, which are this group of nuclei here, which are not necessarily very highly relevant for the talk today, but there is, you can see a lot of connection between those nuclei. So this is what we call the basal ganglia. Some region that we'll talk about today here are the caudate nucleus and the putamen, which are shown here. These are these yellow region here. Here is a cross section of the human brain, and you see though this is the putamen and that's the caudate nucleus. So we'll talk about them a little bit more detail. And here's the thalamus. That's another brain region here that we'll talk about in this presentation. But as one thing to notice here, what happened is that the way that these brain region process information, and so what happened is that information is coming from different regions of the cerebral cortex. It can be here shown as motor, but it can be also associative or cortex related more to emotion and so on. So this information flows into the basal ganglia. Then there is a lot of processing going on here. So it gets first in the caudate putamen, and then it's processed across those brain region to finally leave the basal ganglia to go to the thalamus, which is shown here. And then from the thalamus, it goes back to the cortex. So here, this is what we call the cortico-basal ganglia thalamo-cortical loops, okay? So information flows from the cortex to the basal ganglia, get out of the basal ganglia, go to the thalamus, and go back. And you have a series of loops like that in this circuit that are controlling various aspects of motor and non-motor function. It can be really, as I mentioned, it can be motor control, but it can also be areas that are related more to cognition and also more to emotion. So there are different loops of information like this. Okay, so but that's a classical thing. You can imagine those connections going on that regulate those different aspects of behavior. And here the same thing is shown a little bit more really kind of schematically. So you have the cerebral cortex going to the striatum. The striatum is the composite of the caudate nucleus and the putamen that I mentioned before. So cerebral cortex going to the caudate nucleus and putamen, going to the basal ganglia nuclei, is going out of this to this basal ganglia output nuclei, go to the thalamus, coming back. Then you have these circuits that is processing some information. The cortico-basal ganglia thalamocortical circuit. So what I'm talking about today will not be this circuit. So I'm not going to refer to that. So we do a lot of study on this circuit, but today we're not going to talk about this. So what I'm going to talk about is the thalamus, in addition to go to the cortex, also have a direct connection to the striatum, to the caudate nucleus and putamen. So this is what we call the thalamo-striatal projection. So that's coming from the thalamus, is going to the striatum. So that is the projection that I'm going to discuss with you today. And in particular, one question that we are interested in, we have been studying this system for many, many years, and I'm just going to give you a glimpse today about some data that we have about it. But mainly, the one question that we are currently interested in is trying to investigate whether degeneration, loss of neuron in the thalamus that give rise to this projection, degenerate in Parkinson's disease, may be responsible for some of the early cognitive impairment in Parkinson's disease. And I show you some evidence why we believe that this may happen. So that's really the system, thalamus-striatal system. And the question is it possible that the degeneration of this system may contribute to cognitive impairment, early cognitive impairment in Parkinson's disease? Okay, first of all, let's just give a few words about the anatomy of that system, the thalamus-striatal system, coming from the thalamus to the striatum. So first of all, this is a system that it's not a new connection. It's not a new brain connection. It has been known for a long time, okay? You see here some classical neuroanatomical paper that describe this connection in the early 1940s. So that dates back a long time ago, before any type of modern techniques were available to trace brain connection. People at that time were making lesion of the brain to trace connections in the brain. So this is what those people were doing. But these were classical papers in the field where people were using the tool and traced with this technique, which are a little bit archaic today with the tool that we have, but still were able to identify that there were some neurons in the thalamus that project to the striatum. So this was the thalamus-striatal connection. And you see them. So this is not necessarily a new pathway. On the other hand, even if the anatomy, the knowledge about the connection exists, has been known for some time, what remains unknown for a very long time and is still under investigation is what does this system do? What is the function of that system? How does it relate to the role of the basal ganglia in controlling movement, for instance, and so on? So this is why this system has been somehow known anatomically, but somehow it stays without much attention because we did not know what it was doing. And when you don't know when these connections are doing, somehow no attention is paid to them. But there has been research now since, let's say, the early 2000s that have generated a lot of interesting data suggesting that this system has something to do with cognition. And this is why it has kind of revamping the interest in that system. And also generating, and also the fact that this system degenerate in Parkinson's disease now bring us to think about, oh, maybe this system has something to do with cognition in Parkinson's disease. So just here's an evidence, just one slide on the anatomy of that. How do you demonstrate that this connection exists? So for those who may not be familiar with these techniques, so what we use to trace connections in the brain is we use tracers, okay, which can be of different flavors. They usually are proteins that neurons will take and will transport along their axons, and they will, feel the cell bodies. So you can have retrograde tracers. That means that these are tracers that go from the terminals to the cell bodies, or you can have anterograde tracers that go from the cell body to the axon terminals. But with these tools, with these different tools, then you can trace connections in the brain and you can identify neurons and network that really are related to specific function. And this is how, you can really identify, for instance, this thalamus-striatal system. Here is and the one set of experiment that I've done, it was during my PhD studies a long time ago here. But what we did, then we simply place an injection of this tracer, what we call the wheat germ HRP or Swedish peroxidase, into the striatum, in the caudate nucleus and the putamen. So on one side of the brain, we place an injection in the putamen, to show here. And on the other side of the brain, we place an injection in the caudate nucleus. And these are retrograde tracers. Then we look for the distribution following this. We let the tracer being transported, and then we looked in the brain where we can find cell bodies that were labeled. And these cell bodies were considered as neurons that are projecting to the striatum. And one area that there are a lot of cell bodies labeled is the thalamus. Okay, and here is a schematic of a cross-section of the thalamus, of the monkey. This is all in monkeys. Okay, and the dots indicate these neurons that have been labeled with the tracer. Okay, So what you can see, there are dots in different, and all the name here, the abbreviation of different thalamic nuclei. So two nuclei that we're going to pay attention today here are these two here. The rest, you can forget about that for now. It's not important for the purpose of this presentation. But pay attention to these two here. What is called the CM, which is the centromedian nucleus, and the other one is PF, which is the parafascicular nucleus. So when you look at the cell bodies, you can see that on the left side, which is this one, you have a lot of cells in the centromedian nucleus, whereas on the right side, you have a lot of cells in the parafascicular nucleus, okay? So remember that on the left, we injected the putamen. On the right, we injected the caudate. So what that is telling us is that the centromedian nucleus is a major source of thalamic inputs to the putamen, whereas the parafascicular nucleus is a major source of terminals and input to the caudate nucleus, okay? So the centromedian and parafascicular nucleus are the two main sources of thalamic input to the striatum, okay? And they are differential, they are differentially organized. One of them, the CM, goes to the putamen, and the PF go to the caudate nucleus. Okay, but these are the two main sources, okay, of the thalamostriatal system. So this is this projection that I'm going to talk about today. Okay, I'm not going to talk about this other neuron that you find here in other thalamic nuclei, which is a different story, a different idea. But I focus on these two nuclei, what we call the CM and PF. Okay, so I mentioned to you that the anatomy, then really again, what I just showed you was known already. So this is the kind of thing that these people have identified even in the 1950s. So that was not new. We can refine that and study that in more detail. What about function? And this is where it became interesting with this study that had been done in that regard. So again, these nuclei have been the thalamus as a whole, and this particular group of nuclei have been known for a long time and have been studied in the brain, and their function, their idea about what they do has been somehow there were some ideas put forward. And some people who have been contributors of that I think this is a paper here in 1949, okay? And here we go in the 1950s. Okay, Moruzzi and Magoon, two electrophysiologists, and Herbert Jasper was in Montreal. So these people, what they did is that the idea they proposed, okay, based on lot of different set of data, including, for instance, making lesion of the brain and looking at the activity at the level of the cerebral cortex. So they came up with this idea that there are centers in the brain which are involved in maintaining the state of vigilance of the brain, maintaining essentially the cortex, the cerebral cortex active, and they are involved, then they can be very important in various aspects of attention, consciousness, sleep-wake cycles. So all of those are critical there. And a large part of that is completely true and is still really very valid today. And here is the system that they call the ascending reticular activating system. So this system originates in an area of the lower brain that we call the reticular formation. Then it goes to the thalamus, and from the thalamus, it spread into the cerebral cortex. And in the thalamus, the principal nuclei, the thalamus is a very big mass of gray matter that contains many different nuclei that do a lot of different things. But specifically for that, the group of nuclei that were involved in this aspect of consciousness and vigilance are the two thalamic nuclei I talked to you before, okay? The centromedian and parafascicular nuclei. These are the nuclei that are involved in this aspect of attention and arousing, essentially arousal, and maintaining the cortex in activating the cortex broadly in the state of awareness and vigilance. So this system was known that at that time, this is the way that we are seeing this system very much involved into this type of function. And this was confirmed using more. Now, obviously, this was done in the 1950s, as I mentioned, really making lesions of the brain in animals and doing some EEGs on the cortex and so on. But you can also see the same thing using more modern technique like brain imaging technique that allows you to visualize blood flow in the brain of people, where you put people through different tasks. And these tasks can be very simple or can be more complex. And then that's the way people can identify, using this imaging technique, which part of the brain is involved in what type of function. And here is a paper that appeared in Science in 1996. And it's a very simple story. There's nothing here very complicated. They just ask people to essentially be quiet, and you have the different group they have here. Be quiet, just at rest. And they were measuring using this imaging technique, the blood flow, changes in blood flow in different brain regions when the people were going through brief, sensory tasks. So they were simply either really the people were just quiet there really in the scanner, and there was a sound that was coming, like a click, or a light that was somehow lightning over their eyes. So very simple sensory stimuli, without any consequence, without any, not necessarily linked to any reward or any type of, but just the sound, just the attention that the people were paying to that. And when they were doing this, they realized that there are two regions of the brain that show up and show a change in blood flow. One of them is in the reticular formation, which is shown here, and the other one, indeed, is in the thalamus, and particularly in the central median nucleus of the thalamus, which is shown here. So this is just really a confirmation here, using more modern technique, imaging technique, that indeed these neurons, the cells in these brain regions, are very sensitive to anything that is general attention, okay? So something that you pay attention to. When you have a stimuli coming into your environment, these cells will respond to that. Another study that was done in monkeys this time, using electrophysiology. So then what you do in electrophysiology, then you go with an electrode, okay? You can record activity of cells, okay? You can record neurons are going to change their activity. You can record their activity. You can record action potential in neuron. So what they did in this study, they have the monkey seated in a chair here, and the monkey was looking or hearing a sound, or looking at a little panel here when he was seeing a red dot that was coming in and out, okay? And during he was just looking at these different, or hearing these sensory things, okay? They were recording the activity of neuron in the central median and the parafascicular nucleus. They had electrode in the brain of this monkey, and they recorded to see whether or not the neuron were changing their activity in relation to this stimuli. So what you see here in the diagram at the bottom are the neurons that they have recorded. So each line indicate a cell that responded to these sensory stimuli. And this is the nuclei here, the central median, the parafascicular nuclei. So one thing you can notice, there are blue and red line here, okay? So they identify indeed two populations of cells when they did this recording. And the difference between the two populations, both of them were sensitive to these sensory stimuli. The difference between the two is that one group, those in the central median nucleus, took a long time to respond to the stimulus. So you see here, they responded after about 200 to 250 milliseconds after the stimulus came, okay? So the stimulus could be either a little light here or a beep, for instance, but the response of the neuron came, but it came 200 milliseconds is a long time in the brain. There's a lot of things that can happen during that time period. So they called them, but they responded. And they called them the long latency facilitation neuron. okay? Long latency. The latency is quite long here. They found neuron also in the parafascicular nucleus that also responded to that. But these neurons responded more quickly. So here you see the latency is around 30 to 90 milliseconds. So it's a much quicker response. But both of them had a common feature that they were sensitive to these attention-related sensory stimuli. So all of this goes in the same line, right? These neurons are responsive to these attention stimuli. So what happens if you make a lesion of this system then? And this is what these people have done here. It's a group from Japan who did this study in 2011. So what these authors have done is that let's lesion the system. Let's lesion this projection coming from the thalamus to the striatum and see what happened to cognitive behaviors. So they use a technique that allowed them to lesion selectively this projection. So at the end, they had animals that were where these neurons that give rise to that projection were killed, and they could then test the animal in different cognitive testing with or without the lesion and see what was the effect of the lesion on the behavior of the animal. So here is some of their results. So the type of cognitive tests that they did, these are mice here, okay? We are in rodents, okay? These are what they call these visual discrimination, two-choice visual discrimination task, where the animal essentially is facing two different, really, let's say, levers, and he has to, if he press on one lever, he can receive a reward, and if he press on the other, he does not. So the animal has to learn, which lever is good for him, and then when you learn that, they will press always on the same lever to get their reward. So if you do that in animals that have to train the animals, if you make the lesion of this before you start the training of the animal, okay? So because obviously the animal has to learn that. So if you make the lesion and then train the animal, this is what we call the acquisition. So what you can see here, all those lines are one group, the black triangles here, are the animals that have the lesion of the nucleus, okay? So what you can notice, and here is the number, the percentage of correct response. So what you can see is that the animal with the lesion are about around 50% correct responses. So that means that essentially they're not performing. They go randomly. It's 1 out of 2. So they're not very much higher than just by chance, essentially. On the other hand, if you have animals that have no lesion or controlled animal, they will learn the task and eventually will get to about 80% really success in this task. So somehow the lesion of this system interfere with the acquisition of the task. They could not learn the task. Then they did another experiment in which they trained the animal to learn the task. So the animal got here, and then they made the lesion. Okay, you make the lesion after the animal have learned, and then after that, you put the animal again into the same task and see if the animal is able to perform at the same level as he was doing before. And you can see that they cannot, okay? So you can see that here. So this is the same thing. The triangles are the animal with the lesion. So again, with the lesion, they are, around 50% or maybe 55%. Whereas when they have no lesion, okay, they perform much better. So somehow the lesion interfere with both the acquisition of the task and the performance of the task, okay? So both of them are ideas that this is interfering with some aspect of cognition in performing this task. The animal could not learn anymore what he had to do to receive this reward. And they did some control experiments to demonstrate that this was not due to any motor defect in this animal. So these animals are not making mistakes because they cannot move properly. And these are just experiments here. We don't have to go in detail about them, but these are just experiments to demonstrate that these animals are perfectly correct in locomotion. They have no motor problem. So this is not related to a deficit in motor control. It's really something related more to cognition, where the animal has a problem to learn the task. So to summarize this part, then what that means is that, so what we can say is that the CMPF striatal system, okay, plays an essential role in the process of attentional orienting to external events, okay? So meaning that essentially when you have a sensory stimulus coming in, these neurons are going to respond to that. They're paying attention to these salient sensory stimuli. And we also show that the lesion of this CMPF complex does not induce any significant motor disturbances, okay? Which is something important as well, because as you will see, if these neurons degenerate in Parkinson, you would think about motor defects. Okay, let's talk about Parkinson's disease and the potential role of this system and what happened to this system in Parkinson's disease. So a few words about Parkinson's disease for those who may not be familiar with that. So as you probably have seen already, people who suffer of Parkinson's disease. So this is a disease that dates back from some time. It was identified first time by James Parkinson's, okay, who was a physician and geologist living in London, who identified patients who had different motor problems, okay? And so what are the motor features of Parkinson's disease? So people may have tremor, unilateral tremor of the legs or the arms. They will also have akinesia, which is a freezing. So somehow their muscles are frozen, so they don't move very much. They also have bradykinesia, which is slowness of movement. So everything gets slower. Take a long time to move things, move their legs, move their arms, very difficult. Their muscle also become very rigid, very stiff. So if you try to bend, the arm of someone with Parkinson, you will find this very high rigidity in their muscle. And they have all kind of other problems, motor problems, handwriting difficulties, and they also have a problem with postural instability. So in the sense that they have a hard time to maintain a proper gait. So they will have, and often that's a problem that will make people with Parkinson fall and injure themselves because they have a hard time to maintain their posture. So what we do with Parkinson's disease, how do we treat these motor symptoms? In fact, in Parkinson, it's in comparing Parkinson to other neurodegenerative disorders, it's in a relatively good shape for some time, in a sense that we can treat those symptoms, okay? By replacing the neurotransmitter which is gone in the brain of Parkinson patient. So what is gone in Parkinson patient, among other things, is the loss of dopamine cells, okay? So you have these neurons, and here's a cross-section of the human brain. This is a normal brain, and here is a brain of someone who died with Parkinson's disease. So these black cells here, these dark neurons, are the cells in the brain that contain dopamine. They are found in a brain structure called the substantia nigra compacta. That's the S and C here. So it's pretty obvious that when you have a brain of a control versus a Parkinson patient, you see in the Parkinson patient, these neurons are completely washed out. Okay? So these cells die almost to really 80, 90%. And you can also find, if you use imaging technique, then you will find a loss of dopamine in the brain region that receive projection from these dopamine cells. And this is what is shown here. So all these light here, this kind of colors, indicate the level of dopamine, baseline. There is a high on both sides. But look at that here. So someone with Parkinson's disease, okay, 15 months, look at the lower side on one side here. And then a bit higher, longer period of time, it's even lower. So you can look, this is using here imaging technique in people, alive people, just looking at the imaging of that. But the bottom line is that you have a loss of dopamine, okay? So you lose this neurotransmitter. So what do you do to take care of that? You give them back dopamine. And every patient who come and are diagnosed with Parkinson's disease and have motor symptoms, the neurologist is going to give them a drug which is called levodopa. Okay, the name of the drug is Sinemet, which is the commercial name. But essentially what it is that it's a precursor of dopamine. You take levodopa, it gets in your bloodstream, It gets to the brain, it is converted in dopamine, and then replace the dopamine that is lost, and the motor symptoms will go away when they do that. And they will take this medication every 6 hours, and maybe for some time, they will do pretty well. But Parkinson is not just motor. So what we see from the outside are the motor problem. But Parkinson's disease is a complex disease. Like many of these neurodegenerative disorders, it's very complex. It affects many parts of the brain, and it creates a lot of other problems than motor problem. And here you have some of the other non-motor features of Parkinson's disease that people are complaining about. Lots of problem with sleep, okay? Problem with autonomic dysfunction, okay? Constipation, loss of olfaction, problem with their maintaining their blood pressure, okay? And they also have, some of them will also experience some psychiatric problem. Very common to have depression in Parkinson. Anxiety is very common. Psychosis as well. So these are all really non-motor deficits that are, some of them involve dopamine, but some of them do not involve dopamine. They involve other systems. And you also have cognitive impairments, okay? The cognitive impairments are also complex. You can have some of them that are very early in the disease process. They can come very early, and others that come late. And they probably, the late versus the early, are probably mediated by different neural networks, okay? And usually the problem would be with attention, okay? They have problem focusing on things. They can become easily distracted. And also what we call behavioral switching. So which is also an issue with attention, is that what that means is that they have problem to switch behavior when in response to changes in their environment. When they're doing one thing, they will not automatically reorganize themselves or change their behavior if something happened. They have to think about it. Then that takes time in doing that. So this is what we call behavioral switching. And executive dysfunction, which is maintaining information, store information as they perform a particular task. That means that it becomes difficult for them to do complex cognitive tasks. But they have really a whole panoply of cognitive impairment. And this is what we believe maybe the lesion of the thalamus might be involved into this. And why we believe that is because in the brain of Parkinson's disease patients, so people who died and had Parkinson's disease, neuropathology analysis have shown that the neuron in the central median and parafascicular nucleus degenerate. Okay, so when you look at this is a section here of the brain of someone who died with Parkinson, and this is the central median and the parafascicular, and you can look at the cell bodies at the neuron, and you will find that there is a profound degeneration of cells. And you see here the total number of neurons in these nuclei, the CM, the PF, the kind of the pink one here is the control, and the black one are the Parkinson's disease patient. So you see here you have a loss of about 30 to 40% loss of cell in these nuclei. And the interesting thing here is another scale where you have also the number of cells from these two nuclei. And here are the stage of Parkinson's disease. So Parkinson's disease is a progressive disease, right? And it will go from different stages. We start relatively mild with mild motor impairment. Then we get a little bit more severe, and finally to end at a stage 4-5 where the patients are very severely affected. So here what you can notice here is that the loss of these neurons is not related to the severity of the disease. So if you look at stage 2 and 3 where patients are relatively not too much affected but moderately affected versus someone who is very severely affected, the number of cells is the same. Patients have already lost their 30-40% of neuron, even in early patient with Parkinson's disease. So meaning that this is a loss of neuron that happened quite early in the progression of the disease. So it's not a late stage deficit. It happened early in the disease process. And that's important because we talk about early cognitive impairment. That's important to keep that in mind. And there is a whole literature on evidence for mild cognitive impairment in Parkinson's disease. Okay, cognitive impairment related to executive function are commonly seen in early diagnosed Parkinson's disease patients. So what we mean by that is that these are people, early diagnosed mean that these are people who have just relatively mild symptoms of Parkinson's. The motor symptoms are relatively mild, but already at that stage, they have some cognitive impairment. We do not understand what is the source of that. Why do they have this problem, this cognitive impairment? This is still something which is a very heavily studied in the field of Parkinson. So one possibility that we put forward here that we are studying in our laboratory is, could the degeneration of this system, the thalamus-striatal system, account for some of these early cognitive deficit in attention and behavioral switching seen in Parkinson's disease patients? So this is a study which is ongoing. We have a few data that I can show you, but we are this is something, a project which is moving forward. So how do we do that in monkeys? Then we will do more or less the same approach as what was done in the mice that I showed you before. Is we're going to make the selective lesion of these projection going either to the putamen or the caudate nucleus using this approach here that we don't need to go in detail about it, but that will allow us to kill neurons that project either to the caudate or to the putamen and look at the impact of this lesion on the cognitive performance of these monkeys. So here are some preliminary data that we have from 2 animals so far. So it's an N of 2, it's just starting, okay? So this is relatively complex and busy slide here, but the bottom line, I think we have 2 monkeys, which are called monkey 1 and monkey 2. So here are the data that we collected. So we put this monkey through a series of cognitive testing. So going from relatively simple cognitive task, okay, to more complex tasks that demand much more, integration. And what you can see here on the y-axis is the number of trial that the animal will have to do to reach the criterion. And our criterion is that the animal has to be at least 90% correct in his response, okay? For over 10 trials, the animal has to be 90% correct. Once he reaches that criterion, then the animal go to another task, okay? So what you can see then really with these 2 is that you can visualize that the monkey 1 and monkey 2, let me see here, tries to create, okay, the error. So the blue bar indicate the number of errors to the criterion, and the red bar are the number of trials that the animal has to do to reach criterion. So first of all, we start with something relatively simple, which is simple discrimination. So the animal are shown different shape, and they have to, they will be trained, they will learn that one shape is giving them a reward, so they have to go on the screen and touch this shape, every time it comes to get their reward. So they learn that pretty quickly, and you can see here that the number of trials to reach that is pretty, they don't take a long time to learn this. Then you can really, you can do a reversal in a way that you can, instead, for instance, of having, like maybe in one set of trial, the circle would be the reward. Then once they change that, then you can reverse the other and then somehow train the animal to learn that the square now gives you the reward. So the animal has to learn that and readjust himself. And they do very well on that. There's no problem. Then you can change the shape. You can have different shape, and they will also do very well. They will readjust very well. So then after you have done that, then you move to a more complex set of pictures, in a sense that you still have different shapes shown in green here, but then you start adding these distractors, which are these yellow line here over that. So the yellow line in that case, for this particular compound discrimination, are simply distractors. So the animal can get distracted because they have those lines. Maybe they will not concentrate enough on the shape, and they will start being distracted by those lines. But indeed, in normal monkey at baseline, this is not a problem. They do very well. They will learn this. They will focus on the shape, and the line is not going to bother them. Then you move it again a little bit further, where you start, we talk about intra-dimensional shift of the test, And again, that's more or less the same thing here, in a sense that the animals still have to pay attention to the shape of the green thing without. So again, in that case, they do well. There's no problem. It becomes a little bit more complex then when the animal has to do what we call here the extra dimensional shift. So in that case, the shape may be changing, but also they need now to be trained to pay attention to the line inside the shape. So instead of looking at the pentagon here or the star, what is important is the yellow symbol inside those shapes. This is when they will get their reward. And that is a bit more difficult, even for normal monkey. You can see that they take a little bit more time, but they do it. They will succeed to learn that. So you see, this is a battery here that assess different aspects of cognition in these animals. And at the end, really, a normal monkey will be trained and they will learn that and they will do pretty well. Then what happened is that what we did is we trained this animal at baseline. They all learned this. Then we went and we injected these animals with the neurotoxin to kill the nuclei, and we brought back the animal into the task to see how they will perform into that. So we had two monkeys. So one of them is a sham monkey that we did not lesion the nuclei. We did the surgery, but we did not lesion anything. And the other one is the one that underwent the lesion. And then we did, in this case, we did not do all the basic tasks, but we did mainly the extra dimensional task here, okay? Because these were the more complex tasks we brought them. So you can see the monkey number one, which we see the sham, simply went back on the task and did as well as he was doing before, even better. So it did not take a long time. He had already learned this. He knew what he had to do. He went back and did that. And when you did the reverse, you reversed the line, and he learned that very quickly too, so did very well. On the other hand, when you look at the monkey number two that underwent the lesion, he had a hard time doing this. So he did the first thing, so he was able to do the extra, he learned that finally. But when you did the reversal, so the reversal means then that he had to learn that another set of line was important to respond. So he could not switch there. He could not learn that new behavior. So the red here goes all the way up to 1,000 trial, and even in the 1,000 trial, he did not succeed to make it. So meaning that he had a problem to somehow switch his behavior and be able to learn that he had to respond now to a new set of criteria, okay? So what does that tell us so far? So I think what I've been telling you through this presentation is that the central, median, and parafascicular neuron in the thalamus are the primary source of thalamic input to the striatum, okay? The project to the caudate, nucleus, and the putamen. If you make a lesion of this system, and that we showed that the mouse work that I've shown you from the Japanese group and also some of our preliminary data, may affect what we call cognitive flexibility and behavioral inhibition. So the animal, cognitive flexibility, the animal cannot switch his behavior. He cannot learn that he has to now change his approach to the problem. So that's cognitive flexibility. And behavioral inhibition, that goes about the same line. So they maintain doing the same thing. They keep maintaining the same behavior, even if they should change it. Degeneration of the CMPF neuron may contribute to early cognitive impairment in attentional set shifting, which is more or less the same thing as cognitive flexibility. This is one problem that the Parkinson's disease patient have. They have a lot of time, a lot of problem to, they are not flexible of switching behaviors. So if that is the case, really, idea if we could somehow find a way to protect this neuron from degeneration, that could reduce could alleviate essentially some of these early attention-related cognitive deficit in Parkinson's disease. So that's really the ultimate goal of this whole project that is ongoing in our laboratory related to that, is to identify the role of that system in cognition, whether or not these neurons, the lesion of that system has something to do with cognitive impairment, what sort of cognitive impairment, and whether it contributes, to Parkinson's disease early cognitive impairment. So that concludes my scientific presentation. So again, I want to thank you for your attention. I hope you learned a little bit more about this system, and also all my team here and additional colleagues that are not shown here, in particular Dr. Bachevalier here, who's a colleague of mine who does cognitive neuroscience. So she's the one who collaborates with us on that, and in fact is very much involved, has been designing those tests for our monkeys. And obviously, we thank the funding agencies. Without money, we cannot do research. So we are very grateful to have been supported by the NIH in the United States, also some other private foundation, and and also another foundation here in Canada that we collaborate with. So thanks a lot for your attention, and I'd be happy to answer any question you may have.
Okay. Thank you, Dr. Smith, for that wonderful talk. I really learned a lot about, I actually always thought about the thalamus in a different approach. So I have learned something today. Thank you. And
Good.
I'm going to let the students have questions because I have a couple, but let's see who, if anyone wants to submit a question to Dr. here. Yes, we have one. So it's one of our undergraduate students, Dr. Smith. Cielo says, so in theory, people with CMPF lesions are not able to learn that eggs gives them a reward. Basically, they can't become addicted, or can the role of mesocortical limbic pathway can overpower this effect?
Oh, interesting. So linking that with addiction, whether or not that's a good. So this system clearly, I mean, has never been, but like in fact, looking whether or not this lesion of this network would have something with addiction is not clear. It has never been studied. The bottom line that there's no answer to that. There's never been studied. In general, I think people, when you study the addiction, often we are focusing, I think, as mentioned, on the mesolimbic system. It's focusing on the prefrontal cortex, on the accumbens, on the ventral tegmental area, all these networks. And that's the network that are clearly involved. But, whether having some issues here about like behavioral inhibitions and problem with flexibility might also contribute to that. It definitely could be. So, but this has never been looked at, even in animal model. It has not been studied. So there's no clear answer to that. But it surely does not mean that it just simply has not been looked at.
Mm-hmm. Good. Any other questions? Let me wait. Well, oh, there's another question. Is the loss of flexibility seen in normal aging, not Parkinson, is related to the loss of these neurons?
Yes, it is. That's a very good question. And in fact, with aging, as we age and we start really noticing that pretty early, is that, you have some cognitive changes, right? I mean, happen, including possibly cognitive flexibility, including working memory, executive dysfunction. You're not as sharp, at least the majority of many people. I mean, you become a little bit less, and that's the same thing in human as it is in non-human primate. If we take aged monkeys, they will be able to do things, but they're not performing as well there. So all the different, cognitive function, if you want, that you can think about will be somehow to a certain degree affected with aging. It may not reach, on the other hand, with normal aging, these changes, that's the difference between someone who is affected with a disease versus normal aging, is that these changes do not reach a level where it becomes disruptive to your quality of life and your daily activities, right? And that's the big difference. And in fact, if you take really either Parkinson or in fact even Alzheimer, for instance, even more, Alzheimer's disease, we all have problem with cognitive decline as we age, but in the case of Alzheimer, things get worse and worse and much beyond what the normal aged person will be affected for. And that's the same thing for this. So I would say then the answer to that, yes, clearly our cognitive flexibility is not as sharp as it is as we get older, but normally it does not, if you're not affected with one of these disorders, it will not reach a level where this is going to impact, our daily life.
Okay. Now, I have a question.
Yeah.
I wanted to ask you, I think that the thalamic striatal connection is glutamatergic. Is that correct?
Yes, it is. Yeah. Yeah.
Okay. So then if it's a glutamatergic input toward the striatum, and then the connection from the striatum to the thalamus, is it GABAergic or is it it goes?
Yeah, the striatum to the thalamus, which goes, in fact, through these other nuclei, right? It has to go to these different nuclei, like the globus pallidus and so on, which I did not describe. But indeed, yeah, what is coming out of the basal ganglia is GABAergic. Yeah, it's inhibitory. Yeah.
See, because then the fact that when you lose the glutamatergic connection towards striatum, then I foresee cognitive and learning disability because of the role of glutamate, of course, on long-term potentiation and synaptic plasticity. So that is really very valuable to see how that relates to Parkinson's. So when you look at their circuitry, because when it's coming from the substantia nigra, you're losing dopamine. And of course, dopamine is regulating the inhibition that GABA is also inciding on thalamus. So this is very interesting how the interaction, the neurochemical milieu that is eliciting these deficits, and especially the behavioral switching, because that is also very important for the striatum, of course. The striatum is such an essential part of the brain for changing behavior, the basal ganglia itself. So your thoughts on that?
No, exactly. That you're perfectly right. I think it just makes perfect sense. So I think one question that obviously these are all these complex cognitive behaviors, right, are involve different regions. So we talked today about this thalamus-striatal system, which we believe has probably something to do. But like in anything in the brain, right, when you have a complex behavior which is affected, it usually not only related to one thing. It's a composite of different networks that are dysfunctional. And clearly we know, for instance, as you mentioned about the glutamatergic system being defective, glutamatergic dysfunction in Parkinson's disease is very, very important. But usually the way it has been looked at is because these are changes that happen from the cortex, because the cortex is a major source of glutamate in the striatum, and the cerebral cortex, as you know, is modulating everything, right? So motor, non-motor, cognitive, depending on where it's coming from. And these connections are also affected, in Parkinson's disease. So what is the link between the degeneration of the thalamus-striatal system versus changes in the corticostriatal system in mediating some aspect of cognitive impairment? Is another question that we don't have the answer to that, but you can look at that at different network that are likely contributing to an aberrant behavior and changes in behavior, which we call here cognitive flexibility or working memory or whatever cognitive function the people are suffering of.
Wonderful. Thank you. Thank you for your answer.
Yeah.
Well, I think we finished all the questions for the scientific talk. So now we're going to, Dr. Smith is going to talk about his institution and how so important the opportunities they offer at Emory.
Okay, thank you very much. So that's another part that so that's my other hat that I wear here. And I'm very, graduate education has been for me something extremely important. And I've devoted a lot of time, and I still devote a lot of time to it, in training students, recruiting students, mentoring, nurturing students. This is something very important. And that's something that we take very seriously at Emory. And we have a wonderful group of faculty and students who have graduate education as the top priority into their daily activities. So I will tell you a little bit about the program. And also, I mean, again, I know that probably you can have, and I hope you will have access to my email correspondence and these slides. So please do not hesitate to communicate with me after this. When you see things and you have any question, I'll be more than happy to provide you answers and also refer you to people who may help you if you have anything related to the program. So there's a graduate program in neuroscience. So that's an estimated, that these are the people who make up this program right now. So currently we have 96 PhD students enrolled in this program. About a quarter of them are from underrepresented minority group. And we have a total of around 126 faculty who are members of this program. And these faculty come from a large number of departments and centers at Emory University. So you see here 22 departments. So the neuroscience program is not a department of neuroscience. It's a program that comprises any faculty who do neuroscience research. So they can come from any department, but as long as they are doing neuroscience research, they can become a member of the graduate program in neuroscience, okay? So which differ from some other institutions where you have a department of neuroscience, where you have neuroscientists, but there is a real department there. So here it's not a department, and it's a kind of an umbrella program that comprises faculty doing neuroscience research, but they come from different parts, different departments. And the reason why they come from different departments, then what that means is that they are in those departments because they do different kind of neuroscience. Like I'm a neuroanatomist, I have colleagues here who do neurophysiology, so we are part of the neurology department. I have many of my colleagues who are part of the psychiatry department, the psychology department, the department of biomedical engineering, department of physiology, and so on. But they all do neuroscience, except in different disciplines of neuroscience. Okay, so let's see a little bit. So just to give you an idea about, I'm talking to you about the program, just to tell you a little bit about what I'm doing in the program, I've been doing into it. So as mentioned at the beginning, I'm the director of the NIH training grant that support the graduate neuroscience program. So we can usually, with this training grant, we support 7 of our trainees in their second year of training. This is what we have had this training grant for the past 25 years. So that's a training grant that support a lot of the resources to the graduate program. I'm a member of the admission committee, the executive committee, and the curriculum committee of the neuroscience program. I'm the faculty co-chair of the neuroscience program diversity, equity, and inclusion committee. I've been very much involved in interacting and recruiting and discussing neuroscience with students really from underrepresented minority group. That's something that I take very seriously and very close to my heart. I've been the director of the graduate neuroscience program for 8 years, between 2004 and 2012. I'm currently directing 2 major courses, and we'll talk about the courses that you have in this program, 2 major courses that are part of the neuroscience program for the first and second year neuroscience students. I've mentored a large number of PhD students who did their work in my laboratory. I've been, in fact, a co-director of one of the BP NGO programs. So we had one at Emory and Georgia State University between 2011 and 2016. And we had a cohort of students in this program who came here, and I was the one directing the part of that program at Emory. I'm attending yearly the ABERTAMS meetings, the BP NGO meeting, and the Society for Neuroscience recruitment event for the graduate neuroscience program. And also we have what we call here an Emory Graduate School Lanny Visitation Day here that invite number of students from underrepresented minority group to visit the graduate school, including the graduate program. So I'm the one who is in charge of this visit when the student come and meeting with the student and providing advice and input about the program to the student who visit the graduate school. So the graduate program in neuroscience at Emory, the way it's organized is that the different program in biomedical sciences belong to this big umbrella, which is called the Graduate Division of Biological and Biomedical Sciences, the GDBBS. So that's the acronym used. So these GDBBS comprise 8 PhD training programs. And you have the title here with their acronym, okay? So they're all related somehow to biomedical sciences. One of them is the neuroscience program. And I put here the website that you can go if you want to get more information about the program. Please visit the website. You will have all the details about it, all the makeup of the program, the faculty, the students, everybody is part of that there. But this is part of this large umbrella that include one of the program is neuroscience. So as I told you at the beginning, the faculty there's a large number of faculty, and they come from different departments. So which make it, the neuroscience program, very interdisciplinary, okay? So interdisciplinary essentially mean that you have people coming from a lot of different, who are able to handle a lot of different disciplines related to neuroscience, okay? And you can cut cross, between disciplines. So what are the main neuroscience discipline that people, the type of research they do in neuroscience at Emory? Some people will do system neuroscience, okay, study large systems, understanding the motor system, the somatosensory system, okay, limbic system, but they do. Some other will be even more in molecular research, either in relation to disease or various processings of proteins and molecules involved in neurotransmission. Developmental neuroscience, a big one. Cognitive neuroscience that we talked about, and I gave you some example there of some example of cognitive neuroscience that is going on, but there's quite a lot of it going on in the neuroscience program. Computational neuroscience is also an important component. We have a strong link we have a joint program between Georgia Tech, which is the School of Engineering here in Georgia, in Atlanta, and the neuroscience Emory. So we have a biomedical engineering program, which involves a lot of the neuroscience faculty, and many of them are involved in computational neuroscience. We study a lot of different brain disorders, going from neurodegenerative diseases like me for Parkinson, many people with an Alzheimer's disease research center, ALS, other disease that people study, lots of psychiatric research as well, anxiety, depression, schizophrenia, drug addiction. I mean, these are major components that some people are studying. Epilepsy also is seizures is also very important for many laboratories, stroke, spinal cord injuries, and also a whole series of genetic disorders that affect the CNS and so on. So as you can see here, so that's a very broad spectrum type of neuroscience that is going on in the different laboratories. And one thing that is very important, this is something that personally I feel the most important for my own career and also for the training, is that a nice thing at Emory is that clinical and basic neuroscience research are fully integrated. So what I mean by that, I think I'm an example of that. I'm a member of neurology department. So neurology department is a clinical department. So we take care of patients with disease, neurological disease. There's the psychiatric department. We take care of people who suffer of psychiatric disorders. But these departments also comprise a lot of people doing basic research as well, and clinical research. And there is, the interaction between the physicians and the scientists, is completely merged. We talk all the time together. We have a lot of opportunities to interact. So which is fantastic for helping, particularly when you do basic science research, being able to translate your research to the clinical field is critical, okay? If you are funded by the NIH, that's very important that you know why you're doing what you're doing and what is the impact of your research on brain disease. It is the purpose of the NIH. So having this opportunity of exchanging and talking with faculty involved in who sees patients on a daily basis is something very important, and that helps doing that. And this is something that you can benefit a lot as graduate student in the program to take advantage of these resources. Just a brief overview of the curriculum of the neuroscience program. So if someone come in the graduate program, what are you going to face, and about coursework, qualifying exams, and rotation. So here I have a list. So what we do is, first of all, the student who are admitted in the program came from a lot of different backgrounds. Some of them may come with a neuroscience major, but this is a minority. Most of them would come from biology. We have people coming in with background in physics, in mathematics, psychology. So the bottom line that people come in with a very diverse undergraduate training. So they're not necessarily ready to jump in a laboratory and do neuroscience research. First of all, we need to teach them about the brain, about the basic of the brain, so that everybody, when they start doing their research, they have this formation that we believe is essential for any neuroscientist. Whatever you're doing, any neuroscientist must do that. And we have these 2 core courses here that are given in year 1 in the fall semester and the spring semester, which cover those basics, okay? Neuroanatomy and system neuroscience and molecular cellular developmental neuroscience. So these are 2 big courses, 7 credits each. You meet every day and you get a very strong basic foundation in neuroscience. Then you also have some professional development courses, seminar that will help you kind of building your career, preparing yourself for your research, learning about the opportunities that you have, preparing your rotation, and so on. We also have a grant writing course that we believe, obviously, doing research, 2 things that you need to be successful in is to be successful in getting grant. You need to be funded to do research, and you need to write papers and grant application well. So we start training the student early in doing this. So we have a grant writing course where you'll be trained and you'll be mentored in preparing grant application. We're also very keen on quantitative neuroscience. We know that this is very important, that you understand and you can design your study properly. So you all go to a biostatistics course and various type of quantitative neuroscience courses. And we have a weekly Frontiers in Neuroscience seminar series where you have faculty coming and talk to you about their research. This is a mix of faculty from the outside and faculty from the graduate program. You are required to do at least 3 rotations before you choose your laboratory. Okay, so these are rotations that you will pick yourself. You will decide. You will find a lab that you're interested in. You will meet the faculty. You will then pick 3 of them and do rotations in these laboratories. At the end of these 3 rotations, normally most students are able to choose and decide which lab they pick to do their PhD study. If you need 1 more rotation, you're not yet set, you're not sure, you can do another one. That's possible. And then you have 2 milestones that you need to go over that you need to cross to be able to continue in the program. One of them is a written qualifying exam that happened in the summer between year 1 and year 2. This is an exam that is developing your critical thinking and be able to think about, this is based on the material that you will have learned in these 2 courses, but then you will have to use this material to put that in designing experiment and thinking about the significance of some experiment that you'll be facing with. Okay, so that's our written quad exam. And then in the fall of year 3, just after your 2nd year is completed, then you will have to defend your thesis project. So you put a project together. You will do that during the grant writing course. You will write your project. You will put that in the format of a thesis proposal, and you will present and defend this thesis proposal in front of 4 or 5 faculty who will question you on your proposal. So you need to pass those 2 exams. If it happened that something goes wrong in one of these exams, that somehow you have a problem and things are not reaching the benchmark, you can appeal to retake it. So you can do one retake for each of those exams if there are some weaknesses that the faculty notice, so that you do well into this exam eventually. So our students are usually very successful and do very well in the outcome are very good. And here are some of the outcomes that we can see that are important for us to pay attention to. So first of all, with the grant writing, a large number of our students, year 3 and above, this is when they will submit their proposal, are funded by external grant from the NIH. Usually this is what we call these National Research Scientific Award, that the NIH provide at the predoctoral level. So about 40% of them will succeed to get this, okay? So we're doing well in the nation with all the different neuroscience program. We are listed as fourth in the nation for the number of students who are successful in getting those kind of grants. Student also, we believe that it's very important for students to publish papers during their training. So overall, I mean, on average, it varies, but overall, people who graduate from the program after between 5 and 6 years, we'd have about 4 to 5 papers that they will have authored during their training. And we lose very few students along the way. So when we admit someone, we will do everything we can to bring the student to the finish line. We will provide you all the resources you need. We will help you. We will guide you. We will try to make sure that you are able to achieve your main goal, which is to get a PhD. And indeed, I mean, about 93% of them so far have been successful in achieving that goal. What do the students do once they graduate in the program? So it's very diverse. I mean, students can do with a PhD. A PhD open you a lot of doors, okay? So this is something very important. A PhD open, a doctoral degree is a degree which is unique, which is very important, and this is going to be transformative for you. So anybody who undergo a doctoral degree, 5 years of doctoral study, you will be a different person. And you will be someone who can contribute to the society with a level of knowledge and critical thinking that very few people have. So keep that in mind when you look at what can I do with my PhD. This is something important. And you will be the one who will decide what you want to do with your PhD and how you want to use it efficiently for that. But there are many opportunities for you after you graduate to take advantage of your PhD to help the society and enjoy and find something that you are good at and enjoy. So let's look here at the percentage. Here, your percentage. Most people who will graduate in our program will continue doing research, okay, in different, but you have here, they will go to postdoc. Some of them will get research faculty positions. Some of them will go and do non-academic research. They may join some pharmaceutical industry, but still continuing in research. Others will also be involved in research, but at the level of research management, for instance, in some companies. And then you have a large panoply of different other type of careers. Education is the big one. So students who want to teach in college, they will do that. They will get their training and they will doing well. Some of them will be in commercialization. Scientific writing is also something important. It's something that there are many opportunities out there for people who like writing, who like really communicating science to the community. There are a lot of nice opportunities out there. And we also have some students who will decide to go to medical school and they will do another degree to add to their PhD, and so on. Patent agent, office technique transfer. So it's just an example here of how wide the opportunities are for PhD. So please keep that in mind. This is very important. You have a lot of opportunities. It's a degree which is important. It's a very thorough degree that you get, but at the end of the day, it's going to be worth it, and you're going to benefit of it for the rest of your life. A few opportunities that are there that I've put a few slides on it so that in case some of you might be interested. We have a summer research program that people can apply and join. So this goes to the graduate school. And here is you can look at the web link here for that. This is a 10-week summer research program. If you get accepted in that, you will work in the laboratory. You will be learning various aspects of writing papers, proposals, presentations. You will developing also, you will start thinking about your professional development, your individual development profile. And you will also be taught about how to apply for graduate school, how to be taught about what are the important things that you need to pay attention to be successful if you want to go to graduate school. And here are the eligibility for that. And again, this is discussed in detail here. Clearly, if some people have interest in coming to Emory for your graduate studies, this is a good program you should apply to. They will definitely, it's something I'm sure that when you apply for this program, they will ask, do you have any, are you interested in coming to Emory? Where, what interest of you? And this is something important. But, and I think even for you, as someone who wants for any school, if you want to apply to that school for graduate studies, try to go there for a summer program. That's clearly a good way to know about the school, about the people, about the organization, about the city. Really, all kind. If you have the opportunity to do that, do it. That's very, very important. And here are some of the things about the GPA, a 3.0 GPA minimum, and obviously they want people who will still have a little bit to continue in their studies and so on. But something to look at. Look at the website. You will have all the information. So one thing that we are very also, Emory in general, and the graduate school and the graduate neuroscience program, we do everything we can to have a program which is a makeup which is very diverse. Okay, so we believe very much in the importance of diversity, in bringing people with different background, different gender, different ethnic groups, people with different ideas. So this is what enrich, our community and our program. And this is something that we take very seriously, and we will make every effort we can to make sure that that happens. And clearly in regard to students, we pay attention to that at the admission level. So if we want to achieve that, clearly we have to have that part as a priority for our admission. And you can see here that is, that's the makeup of the current students in the neuroscience program. About 50% of them are white, but you have then afterwards, about 50% are coming from different ethnic background. Asian, Black, Hispanic, 2 or more races. So we try to definitely make sure that we maintain at least that, as we move forward. Currently, we have a large number of women in the program. So 65% are female, 35% of male. So the women, the girls are doing really well, and they are getting into graduate school more easily. And the cohort of people that we have are mainly female right now in the program. A few tips about admission from here, at least from the neuroscience program, just a little bit of information. So usually the way we have one round of admission per year, usually the due date is early December. This is when people submit their application. Then, a subset of people are going to be, if you pass that first, you are chosen by the admission committee. Then you will come for interview in February. Usually we have 2 weekends of interviews. Clearly this year, because of the pandemic, the students are not going to come on campus. This will all be done virtually. But normally you come here, you spend a weekend, and then you meet with faculty and students. We usually bring in about 15 to 18 students per year. That's about the number of students we have been bringing in over the past many years, okay? And as I mentioned, we try to make sure that we maintain, at least, 25% of students coming from URM group. The success of your application is going to be based on many different things. And that is not only for Emory, it's valid for any school that you will apply to, okay? So see your application. Every aspect of your application will weigh into the balance when come the time of the admission committee looking at your application. Clearly, you need research experience, okay? We do not accept students who have no research experience. Even if their GPA might be 4.0, they will not get in if they don't have sufficient research experience. So research experience is very important. Try to do that, get some of it. Really, this is critical, okay? GPA, we obviously look at the GPA. Okay, we usually above 3.0. This is what I would say as the lower. There is a large here, really, we can see the range here. So student can get in with a GPA between 3.0 and 4.0. Okay, so I would, the GPA, really, yes, it's important. You need to go, at least between that, but this is clearly not the only factor that come into play. Okay, so that's very important to keep that in mind. You need solid letters of recommendation from research advisors. So that's why if you do research, you can get letters from those advisors. Those letters are very important. They have to be meaningful letters. They have letters that will tell us about you, who you are, what you have done in the laboratory, how well you did that. So this is something that we will use to guide us as to whether you're ready for graduate school. Your statement of purpose that you have to write has to be also meaningful, has to be inspiring. We have to find through that, that you are ready for that. You are ready. You're going to graduate school. You have a goal in mind. You're doing that for a it can be different reason why you're doing that. But clearly we can find that you have a motivation and a goal, a career goal in mind. And clearly we want to see also people who are matching with the program, what we offer, right? So if you want to do something in the program that we don't have the resources to provide you, we'll let you know, and you will probably realize anyway yourself that you may not be at the right place. There's no need for GRE. GRE tests, we stopped doing GRE testing for the past couple of years. We do not need those. We do not believe that they are a very good indicator of students who perform in research, so we don't require them anymore. So as I mentioned here, when you apply for graduate school, pay attention to all elements of your application packet. Everything will be looked at and considered by the admission committee. So you need to really take the time and pay attention to everything, because everything is going to be considered. So here's a few words about Emory University in Atlanta for those who never came to Atlanta before. So obviously Atlanta is a big city in Georgia. That's the biggest city in Georgia. That's the capital of Georgia. You have a view here of the downtown Atlanta. The university, Emory University, is located about 4 miles downtown of Atlanta. So it's very easy to get downtown. That's the nice thing about Atlanta. So it's very easy to get anywhere unless you live in the suburb outside the perimeter. But it's about 4 miles the northeast of Atlanta. It's a beautiful campus. A lot of wood, lot of trees, very nice walking campus, very active. And you can see here, you see the campus here, and you see the downtown here. So that's Atlanta, okay? This is what you can see there. Very diverse population. So as I mentioned that before, so this is something that university and we take as a very important thing, and we keep maintaining that. The cost of living is very reasonable compared to other big cities in the US, particularly cities on the East Coast or the West Coast. This is a very, very reasonable cost of living. With the stipend you receive for this, students really can do really well. I mean, in doing their study and not have to work outside to get extra money for that. There's a lot of activities, of course, a lot of nice restaurants, bar, and entertainment. With the big city, you can have access to any type of culture or anything you want. And for those who are interested in hiking, and North Georgia is beautiful. You are close to the Appalachian Trail that go for really long distance. So this is thing that you can get there. It's about an hour drive from Georgia. So I've been here myself for 25 years and moving from Canada, from Quebec. I love Quebec. I go there every year. I love it. This is my home. But clearly we have, both my wife and I and my son, we have a great life in Atlanta. We enjoy it. We love Emory. We love the people we work with. And we like the Southern hospitality. I mean, it's a very interesting, and we really like the way that people interact and and we communicate between each other. Okay, so the last slide here. So what to think about when you think about the Emory Neuroscience program? First of all, this is an interdisciplinary program, so you will receive training and you will have opportunity to approach neuroscience from different angles. Very strong integration between clinical and basic science, very important. That's something which is, as I mentioned, for you, for me, it is the cornerstone of this is the one thing that I feel is so critical and has been so critical for me to be successful here. A lot of collaboration. Collaboration is very easy. People collaborate a lot. Okay, so that's something important too. Collegial and transparent leadership. So we bring the student in the leadership of the program. As a student, you will participate in the leadership. You will have opportunity to sit on all the committees that are leading the program, and you will have your word to say. Okay, that's very important. We rely a lot on students to guide us in adjusting and editing the program as we move through. As I mentioned, we value diversity, equity, and inclusion. It's very important. And this is a very friendly relationship that we have. That's part of the culture, the program, the interaction between the student and the faculty is very, very friendly. And here I just put some information. If you want to have more details, if you want to talk to me by email, my email address is there. I also listed 2 people here for those who are interested in admission in the program. This is the director of admission, Dr. Gordon Berman. This is his email address. And you have here Mrs. Chanel Loiseau, who is the program coordinators. So either of them also can, if you have questions about admission, you can communicate with me or you can communicate with them and they will refer you to the proper place. And then I added a series of links here that you may also want to consider. We also have an MD PhD program here at Emory. That's the website for the MD PhD program. Those of you who might be interested in going into that route, this is a very good, very strong program. Lots of very great students who joined the neuroscience program through this, and so on. So I think that's it. And I know it's almost 5:30. Yeah, we're done. And so I hope this was useful. And I hope to hear back from some of you. And if you have questions, I'd be happy to stay and answer questions you may have right now.
Thank you. Thank you so much, Dr. Smith. That was a wonderful overview of the neuroscience program at Emory. And I feel that it's such a successful and very attractive program for all our students. I think everyone should consider a summer and later graduate school at Emory for sure. Unfortunately, we reached our time to close, so we won't have space for questions. Nevertheless, the information that you have provided us, and we already put it in the chat, so the students can access it as well right now.
Okay, good.
Thank you so much for being here and you know, talking about the wonderful science and also showing the importance of this interdisciplinary program that you provide at Emory. I really appreciate your time and your consideration toward us. And
okay, so, that's great. Yeah, it was a great honor for me. So thanks a lot, and thanks to all of you who stayed there. I hope it was not too heavy, you know, to hear particularly the scientific part. You know, I know that it's we get all of us, and one day it will be you. You get very excited about the science we do, and you get passionate about it. But I hope at least that it was a good composite and you learned something.
Yes, I think we all did. So
okay, thank you
so much. And then we just want to encourage everyone to come back next week on Wednesday because we continue till the end of the year. And this time we'll have Dr. Kelly Monk, which is professor and co-director of the Bollum Institute at the graduate program in Oregon Health and Science University. So we'll now move to the West and listen from Dr. Monk on her presentation next Wednesday. So thank you, Yoland. I really
take care. Have a nice week. Bye-bye.
You too. See you guys later. Bye. Take care. Bye. Bye. Bye, Yoland.